All right, so I'd like to start by thanking you guys for joining me for my presentation, Magnetoreception, Final Frontier of the Senses. So we'll go ahead and jump right into it. So this is a picture of the Arctic tern, and over the course of its life, it'll migrate a total of 1.5 million miles, and this is all thanks to a sense called magnetoreception. Magnetoreception is a pretty dense topic, but hopefully by listening to the presentation, I'll be able to inspire a little bit of curiosity about the natural world in you. And my own curiosity about this topic was inspired in my biology class when we were talking about sensory systems, and I found that there's little to no information in our textbooks. So I decided to look at the scientific literature to try to learn as much as I possibly could about it. What I found was that there's two primary types of magnetoreception. One developed secondarily to the other as a more diverse group of species began to inhabit the planet. And the first type of magnetoreception that I'll talk about is called the magnetite particle model, or MPM. It was first discovered in bacteria, but it is found in all three domains of life. So bacteria, archaea, and eukaryotic cells, which are animal cells. And in this model, you can see from the picture on the screen that there's a magnetotactic bacteria, and it has a chain of magnetosomes. And the magnetosomes act as a mechanical receptor by aligning themselves between the north and south magnetic dipoles, which allows the organism to orient itself. And the other type of magnetoreception is called the radical pair model, or RPM. And this one's a little bit more complicated, but basically it starts with light entering the eye over here. It hits the retina, where it activates this protein called cryptochrome. Cryptochrome adapted to become a magnetoreceptor, and once it is activated, if you look to the right, a lot of really complicated biophysics happen, but it results in a radical pair of electrons that form, and those electrons can interact with the magnetic field to send a chemical signal to let the animal know what they're sensing. And so as I found more and more information about these two types of magnetoreception, I really wanted to look at how they evolved, whether they're connected or independent of one another. So looking at a geologic time scale here, the first organisms that arrived on the planet were archaea, and they arrived over here in the mid-Archean eon, which was about 3 billion years ago, or 3,000 million years, which is the time increment on the scale. And after archaea came bacteria, and then animal cells, or eukaryotic cells. So this, the first type of magnetotactic organism was also archaea. So that would suggest that MPM came before RPM. And so the next thing I did was to build a table of different magnetotactic species that use RPM versus MPM, and then build a phylogenetic tree to look at how they're genetically related to one another. And so looking at this phylogenetic tree over here, the ones that are listed in red are animals that use MPM, and then the ones listed in green use RPM. So what's important is that this tree shows that it's a it's not a monophyletic trait. So what that means is the type of magnetoreception an animal does not follow any single genetic line. If it did, you can kind of see how these break off into different branches. So there's a branch up here, and there's a branch down here. And if it was a single genetic trait, you would have a whole branch in red and then a whole branch in green. But because they're mixed in the different branches, that signals that the trait is selected for by the environment. And that's supported by looking at the environment that these different animals live in. So birds, newts, amphibians, and butterflies and fruit flies all live in environments with a lot of exposure to UV light, which makes sense because RPM is mediated or activated by UV light. And if you look at the animals listed in red that are use MPM, so that would be the mole rats, bats, the trout and salmon, termites and ants. Those are all critters that have low exposure to UV light, so they're burrowing insects or animals. They live mostly underground, or they live in caves and they're nocturnal, or they live on the ocean floor. And in the ocean, at 30 meters of depth, the water cuts the amount of UV light that penetrates the water by 50%. So it would make sense that those different organisms use MPM since they have less exposure. So after I began to figure out how these evolve and that they're independent of one another, I began to look at cryptochrome specifically and how cryptochrome changed and was able to be adapted to become a magnetoreceptor. So one of the first things I did was to look at the evolution of cryptochrome specifically. So they first started, as a photolyase and then evolved into plant cryptochromes and then cry-dash and then into animal cryptochromes. There's many different types of animal cryptochromes and different animals that are magnetotactic use different types of cryptochromes for their magnetic sensing. But what's interesting is the photolyase, commonly found in different fungi. It is an enzyme that repairs DNA that has been damaged by UV light. And in plants, cryptochrome is responsible for circadian rhythm and also for the growth and development that plants undergo in response to light, and then seasonal changes that occur with plants. And then in animals and fish, cryptochrome again plays a really important role in circadian rhythm. And so one of the most important things to look at is the genetic sequence of the cryptochromes that are used by different animals for their magnetic sensing capabilities. So what you're looking at here is 4 different species that are magnetotactic. These are the 4. And then a segment of their cryptochrome protein has been sequenced out here. And it aligned those sequences to see where the sequence is homologous, meaning they're identical, and where they're not. So anywhere you see a star, there's 100% homology. So the genetic code is exactly the same between all 4 species looking vertically. And if there's no star, it means it's not 100% homologous. But what's important is that genes are directly responsible for protein function. So if different animals use different cryptochromes to be able to have a magnetic sensing capability, there would have to be some gene homology in the different cryptochromes because it has the same function. So it should have the same genetic code, and that's exactly what we see here. And what's also interesting is we see the same pattern in the evolution of cryptochromes. So here, instead of looking at the genetic sequence, we're just looking at the length of the protein, which is the length of the amino acids. And so we see that there's 2 different ends to a genetic sequence, an N terminus and a C terminus. So we see that the N terminus is highly conserved, and it's exactly the same length, and the genes that are there are almost identical between the photolyase, between the animal cryptochromes, and between the plant cryptochromes. So that's what allows the shared capacity for the magnetic sensing capability, and why all these different things are activated by UV light. But the different length and the different C terminus that would be the part of the protein that has different function because there's different amino acids there. So that's why CPD photolyase doesn't have anything to do with circadian rhythm, but in animal and plant cryptochromes, they do. And then in plant cryptochromes are even more important because they have a huge developmental aspect. So it has even a longer cryptochrome protein. So that's what was so interesting in seeing how cryptochrome evolved to become a magnetoreceptor, and that we have really strong evidence to show that even though different animals use different cryptochromes, the part that is used for magnetoreception is still the same. And one of the biggest challenges I faced when I was researching cryptochromes was that there's no common nomenclature for the naming and classification of different types of cryptochrome. So in order to have further research, I think it'd be really beneficial to establish a common nomenclature in the scientific community. And even though I had this challenge, we're still able to see a lot of the commonality in these cryptochromes, which was also what was really interesting. So I hope that by learning a little bit about magnetoreception and cryptochromes, you were able to be a little bit curious about the natural world and why things work the way that they do. And in closing, I'd like to give a special thanks to the Honors College at Shoreline Community College, and then Brooks Zimmers and Terry Taylor specifically, and my research mentor, who I worked very closely with, Erica Shara.
I really liked the new